So at the end of chapter 3, I think your author, in this book, it's Anatomy and Research Methods. So at the very end, we have some of the research methods that exist out there. And I'm sticking with this outline that I created from a different textbook, but these are the categories that are basically out there in the world. And so I think it follows really nicely still with CALIT, starting with looking at brain damage, but then getting more specific about the structures that we're looking at by using some kind of ablation or lesioning, then stimulating neural activity, recording neural activity, and then looking at the structure of the living human brain. And I have out here numbers 5 and 6, neurochemical methods and genetic methods. The neurochemical methods, CALIT does not discuss those. And I have another textbook that does a really good job of going through some of those techniques. They are rather fascinating, but I am going to skip over those neurochemical methods. And then genetic methods, why that's also pulled out here, is your author discusses using twins and looking at genetics more in the development of the brain chapter, those genetics, evolution, all of that. He puts all that together. So that's pulled out as well. So we're going to start by looking at damage to the brain. And if we're looking at brain damage in humans, this is usually produced by a brain tumor, somebody having a stroke, maybe some kind of head injury, some misfortune that produces a naturally occurring brain damage. But this, luckily, this is not very common, but for scientists, that makes it difficult to really get enough participants to figure out what this area, what's happening in a particular area, although we still do a lot of work with people who have some kind of brain damage. But the other problem is that it's really not, typically it's not specific to some particular region or area. Brain damage is really quite messy, and so we have these impairments, and it's difficult to say that this is the exact location of that behavioral outcome, as it's difficult to pinpoint the exact location and severity of the brain damage. I start here with the example of Broca's aphasia, which we learned about from Pierre-Paul Broca, and thus the name, or the Broca's area, actually is named after Pierre-Paul Broca. He was working with people who had these kinds of language disorders, and especially so after somebody had a stroke or had some damage to the brain during their lifetime. And probably the most famous patient of his is Tan, who for the most part appeared to understand what people were asking him to do. So his doctors might give him instructions or ask him about certain things, and he could follow instructions and to some extent answer in communication that didn't involve language, because all he could actually say was Tan, which is how he got, this is not his real name, but that's just what he could say, was Tan. So that was pretty severe damage to Broca's area. And then I'm going to ask you, you can either pause here or wait till the end of this slideshow and go watch a bit of Sarah Scott. I think I usually show about 5 minutes in class, but however much you want to watch of her, you can see that she also has Broca's aphasia. And you might think about, and go ahead and write down to think about the question of what can you say about Sarah Scott. And I really, so last semester I got to do this with my students, and I hate kind of missing this, and we might end up coming back to this if we talk about language in the brain, but do notice what you can say about her, or notice about her language abilities and her language inabilities. So what she really lost with this Broca's aphasia. And I will just go ahead and tell you that this was soon after her stroke, but that she's young, and so the brain can handle this damage better when we're young, as it's easier to rewire and develop brain regions, or get back some of those brain regions, and they did a really neat new technique of helping people with aphasia, where they are using that transcranial magnetic stimulation, which we will talk about, to stimulate that part of the brain while asking her to do these kind of language production tasks. So if you look her up after watching the video I give on YouTube, so this one's really soon after the damage, if you look her up, you can see that her mother continues to film her, and her language skills get better and better. And so because brain damage is in people is usually pretty messy, and it's not a specific region that we can point to, or some little, especially when we're looking at some of the small areas within a brain structure, that's very, very rarely a clean kind of damage. So one of the things we do is we do ablation or lesioning on animals, and these are slightly different but very overlapping things. And ablation is surgically removing some particular brain structure in an animal so that we can look at what we now see as impairments and see how that structure plays a role in that particular behavior. But a lot of times what the researchers are looking at are these smaller structures that are quite difficult to remove surgically in this way. And if you'll notice, so if you go back in your mind to that PowerPoint with the hypothalamic nuclei, so they have looked at specific small nuclei of the hypothalamus, which is already a pretty small structure. And so what they're going to do in that kind of case, also with the amygdala, they know little tiny parts of the amygdala, areas of the amygdala, what their role is in something like aggression or whatever, by using these stereotaxic instruments to lesion or damage a specific area of the brain. And you can see the stereotaxic instrument there, that you would put the animal in there, keep the head very still, and then we get a brain map. I'm actually going to go through this in the next slide, so I'll just move along. We get this map of the brain of the species, a stereotaxic atlas, and then so we anesthetize the animal, drill a small hole into the animal's head, and one way to do this is to insert an electrode and pass electrical current. And so this is showing, so those are the ventricles of a rat, those big blob-looking things in the center. And what you can see around that, what those pink arrows are pointing to, are areas where you can just see fewer neuron cell bodies, which is what those little dots are, because those areas have been lesioned in this particular animal. So I'm still in that getting the electrode into the specific region that we want to damage, and passing an electrical current. This is done by passing a very high radio frequency current that is producing heat and killing the cells in the region that surrounds the little tip of the electrode. What this does is it damages everything in the area, including the axons going through the area. So even this is a little bit messier than we want. Sometimes they use these excitotoxic lesions, so usually it's glutamate, and that will, if we're injecting some glutamate into an area, that will produce these excitotoxic lesions, which so the cell bodies themselves are what die. And so we can say this is specifically what's happening in this brain region. And I'm going to talk here also about the importance of sham lesions before I do my mentioning of, so no, I'm going to go ahead and obviously mention, if anybody gets really uncomfortable about working with animals in this way, because this is the really the beginnings of talking about how we utilize animals, because we'll see it a few times. Do let me know. The only thing I can really think to do is to put a discussion out on Blackboard, so that if you want to discuss how you feel about animal ethics, you can. I think there was one person in one of my classes who in this semester that wrote down that that's one of their interests is to discuss ethics of animals. I can't remember which class. I do have it written down somewhere, but do let me know. That is something you can certainly think about writing one of your applied homework assignments on. But if you want to have a discussion out there, that I think it's an important thing to see that different people feel really differently about animal ethics. And I did do a little bit of animal ethics early on, and I think I mentioned that, so I just want to mention it again, that is something we can do and set up. And if people are really interested, we could even maybe use one of those extra days to discuss that. Those days when we have kind of an open schedule, part of the schedule. Okay, so here here it's important to talk about why sham lesions are important, and this is kind of like talking about the placebo effect, but not exactly. That belief in people that if I'm getting some drug, my belief that that drug is going to work can actually influence me. Well, all of everything we've seen so far of before we get to actually doing the lesioning, so that anesthetizing the animal, boring the hole through the head, and getting to the point that we want a lesion, that in itself could be affecting the animal, even without the actual lesioning that we're doing to look at this specific structure. And so it's really important to do sham lesions, to have this control group of animals that have that get anesthetized and have the radio frequency electrode put into their head. And then there's this so they don't actually go in and send the radio frequency current to the brain to kill any of those neurons. And that way we can have a nice control group for those animals. Now we can perform this kind of temporary lesioning on humans as well. So the transcranial magnetic stimulation, I mentioned it for Sarah Scott, that they were enhancing activity at that Broca's area, but we can also use this to temporarily depress activity in a specific brain area. So they use this magnetic coil mounted on a small paddle, and it's going to deliver a magnetic pulse. And this can be a small enough pulse that we're just enhancing that area a bit, but we can have a strong enough pulse that we overstimulate those neurons so that they're not able to be used for a particular task. So we can have someone do a task both as normal without any temporary lesioning, and then temporarily lesion an area and see how they perform that same task if we have some brain area that we think is involved in that task. This only really penetrates about 2 centimeters, so we're talking about what we can do at the level of the cerebral cortex. We cannot really use this at the level of any of those subcortical structures because it's not really reaching that deep into the brain. So we're going to look at a couple of studies here that have done this temporary lesioning. So one, TMS has been used to disrupt activity in the visual cortex, which causes people to have no conscious experience or perception of those visual stimuli. So basically giving them a temporary blind sight experience. And we do see that those people still direct their eyes toward a visual stimulus, so a light. So if you remember from blind sight, while I'm saying and denying that I see anything out there, consciously I can still interact with the visual stimuli. I can kind of navigate with those stimuli and know where things are. Another example is Grossman. They temporarily lesioned this superior temporal sulcus, which is an area in the temporal lobes that allows really for us to see and utilize biological motion. And so this was temporarily, TMS at this area, temporarily disrupted our ability to detect biological motion. So they sometimes put these point lights or these lights on people's joints, turn off the lights, and so all we can see from the person really is their movement. And they can simulate this as well, And I sometimes show a little bit of this in my sensation perception class, that we can really easily pick up not just that this is biological movement, but that we can see when they're doing jumping jacks versus, doing a hula hoop or playing basketball. We can tell what the person is doing just with these. I think it's sometimes between 9 and 13 lights on the joints of the arms and legs mostly, but also in the central body. And Grossman had it so that there were also lights on the screen that were moving randomly, and we can make it so that we can see biological motion even if there's a lot of random lights on the screen moving randomly. We can still see the person within those fake lights, the lights that are moving randomly. We can see the person's movement still, but as you were using the transcranial magnetic stimulation to depress this superior temporal sulcus, those that we couldn't bring those random lights. We had to bring those down and have fewer and fewer of those. For the person could still see biological motion to some extent, but they needed to have fewer of those random lights moving around randomly. Hope that came out making sense. And your author has a different study that he describes, and so it's good I think to have a lot of different examples of how we can use this. We get to number 2 in our outline there, that is about how stimulating neural activity to look at how the brain works. And I have a couple of examples here of electrical stimulation of the brain, which we can accomplish. And I'm mentioning here again, very briefly, that we can use the magnetic field, apply those magnetic pulses to the scalp and stimulate the areas beneath it. So in the transcranial magnetic stimulation, so remember that's at the level of the cerebral cortex. And in order to get to some of those subcortical structures, we can also send a weak electric current into a brain structure through an electrode to stimulate that area, sometimes called electrical brain stimulation, or EBS, sometimes called deep brain stimulation, or DBS. These ways of stimulating neural activity don't exactly duplicate normal signals, because what they're doing is they're stimulating a brain region. And this is what usually happens in the brain, is we have these kind of neurotransmitter or these circuits of brain activity for particular things. So some small circuit within the region is usually what's active when we are performing some task. So one of the ways that is really interesting and new, a way to look at this, is to use optogenetics, which allows researchers to stimulate activity along one of those circuits in a brain region by using a laser light. And my understanding of this is very superficial, and I have this kind of short video that if you add this to the Sarah Scott video at the end of this, that I have run out of time. And just so watch the optogenetics video, and we'll be done until Friday.